So welcome everyone to the My Tutor online school. This lesson is in GCSE Biology and is about transport in cells. It will be 1 hour long, so we'll finish at 4:15 and it will be about 45 minutes of content and then about 15 minutes of questions and past paper questions and things at the end. If you've been a part of the My Tutor online school before, and in particular biology lessons, you'll know what to do with popping questions in the Q&A box and in the chat, but do that if you're stuck. I'm taking biology lessons. My name's Charlotte. I have been tutoring with my tutor for a couple of years. And I studied conservation and biodiversity at the University of Exeter, so I'm a particular specialist in kind of the ecology modules and things. And I now work for a charity called the Farming and Wildlife Advisory Group on a daily basis. Okay, so today's lesson is going to be about transport in cells. It follows on from yesterday's lesson, which was about cell biology. So if you attended that one, then this will follow on nicely from some of the things that we discussed. If you didn't, then no problem. This also stands on its own. We've particularly tailored content to the AQA specification. But if you're not studying AQA, then a lot of the content combines over. We've kind of tried to cover general topics that are present in all specifications, and this topic in particular, along with cell biology, is present in all specifications, so you're definitely in the right place. We'll do some mini quizzes and things as we go throughout, so if you have a look down at the bottom of your screens, there's the question and answer session, and there's also the chat option down there. So if you pop the answers to any mini quizzes and things in the chat, and then any questions about the content, pop that in the Q&A section and we can answer those, and I'll try and do a few sections and answer questions about those sections as we go along. Okay. So last time we talked about cell biology, animal and plant cells. We talked about eukaryotic and prokaryotic cells and the difference between them. And we also talked about specialized cells. So we talked about animal cells, in particular nerve cells, muscle cells, and sperm cells. And we talked about plant cells, such as root hair cells, photosynthetic cells, xylem and phloem cells. We covered all of those and how they're specialized for their function. And a lot of those specializations, we talked about things that link to the transport of substances in cells. So for example, we talked a lot about having more mitochondria to provide energy for active transport. And we also talked about cell membranes and how they allow substances in and out of the cell. And today we're going to talk about how that happens. So we're going to talk about 3 different types of transport of substances in cells. So we're going to talk about diffusion, osmosis, and active transport. Okay. So the first thing that we're going to cover is diffusion. So the official kind of definition of diffusion that you need to learn is the passive random movement of particles, which results in them spreading out from an area of high concentration to an area of low concentration. And in brackets there, I've put down little things to note. So all particles are moving all the time. That's something that we've learned in lessons, probably over the last few years in your science lessons. So particles are always moving. They have a certain vibration and they move in all directions. And that can be affected by temperature, which we're going to talk about a bit more, but so particles move randomly in all directions. But if you've got 2 different types of particles and they're randomly moving, then they will spread out and mix together. And this means that if you've got a group that are grouped down the bottom, as you can see here, they will randomly move about until there's a mixture. And we call this moving from an area of high concentration. So here the red dots are all grouped together in a high concentration of red, to a low concentration. So in this top area of the beaker here, we've got a concentration of blue, but we have almost no red particles, so a low concentration of red particles. And so with that random movement, they will spread themselves out into that area of low concentration. And we describe this as going down the concentration gradient. We're going to talk about this a lot today, so it's well worth getting that straight in your mind. When particles are moving from high concentration to low concentration, they're moving down a concentration gradient. It's fairly easy to think about because you're moving from high to low, so you are going down. So we'll talk about that a lot today. So how can we manipulate that rate of diffusion? So we've talked about diffusion in quite a lot of context within the body, and we're going to talk about that more on the next slide. But we've also talked about how we can change that rate of diffusion. And one of the first things that I mentioned just now was temperature. You can see at the bottom there. When you increase temperature, particles move faster, they have more energy. We know that from physics, and it comes into biology just as much. So increase the temperature, you're increasing the movement of particles, and so you increase the rate of diffusion because they're bouncing about all over the place more. So they're more likely to do that random spreading out. You can also affect diffusion by concentration. So the greater the concentration difference, the faster the rate of diffusion. So in this example, we can see all of the red dots were concentrated at the bottom. So there's a really high concentration of them there and a really low concentration of them at the top. If you had a couple of red dots at the top, there would be less of a difference. There would be a smaller difference in concentration between the top and the bottom. And so there'd be a little, the rate of diffusion, so the spread of those red particles to the top of the beaker, would be slightly slower. Okay, so in a sense, if you're talking about going down the concentration gradient, if you think of that as a slide, the slide wouldn't be as steep. Okay, so that's kind of diffusion as a basic concept. It's the movement of particles, it's how most things happen. It doesn't need any energy, it's a passive process. And so most of the exchange of particles in our body is done by diffusion. So some specific examples of that. So dissolved substances like sugars. So glucose is a classic example of sugar, and we talked about that with respect to respiration yesterday and photosynthesis. So glucose is kind of a food molecule that runs around both plants and animal organisms and is used for growth, for movement, for all of those things. Gases, oxygen and carbon dioxide, again, integral part, respiration and photosynthesis, our breathing, transpiration in plants, and also waste products, urea, which is a waste product from lots of other processes, move in and out of cells. So all of those things, so glucose, oxygen and carbon dioxide, and urea move in and out of our cells on a regular basis. So if we look at that in detail, so diffusion and waste products, amino acids are broken down in the liver and urea is formed. Urea passes from the liver cells to the blood and is then excreted from the kidney. We'll learn more about this later, so don't worry if all of that's kind of news to you. So we do a lot more about that when we cover homeostasis and maintaining water concentrations in the body. So urea makes all of those moves, so from the liver to the blood and then to the kidney, all through diffusion. So urea is really small, it can fit through cell membranes, and it can just diffuse across those partially permeable membranes. Diffusion and gas exchange. If you've been part of the My Tutor Online School for a while studying biology, this is one of the first things that we covered, was gas exchange in the lungs. So we talked about alveoli in particular in the lungs, and how oxygen, when we breathe in, moves from our alveoli into our blood. Carbon dioxide is exchanged for that and then breathed out. All of that is done by diffusion as well. So literally just moving from a low concentration, from a high concentration to a low concentration through that random movement of particles. And finally, diffusion and digestion. So food substances like glucose will diffuse across from the intestine into the blood before that glucose heads round to all of the things like muscle cells and other parts of the body. It can diffuse across the membrane surrounding the intestine into the blood. And to add to what we talked about, those two factors that we talked about that affect diffusion, so temperature and concentration gradient. When we're talking about gas exchange in the lungs or diffusion and digestion, we also talked about three things that affect that rate of diffusion. So within the cells, we talked about how surface area affects the amount of diffusion that takes place, and the alveoli that you can see here in this diagram, we have lots and lots of those in our lungs to increase the surface area where gas exchange can take place, so where that diffusion can take place. And in our intestines, this is an example of a cell on the wall of intestines. They have lots of these villi, these kind of protrusions with little capillaries in them. And those protrusions provide a really big surface area and a really short distance between the food particles and the capillaries on the other side carrying the blood. And so there's a really big surface area and a very short distance for diffusion to take place over. So that all makes it quicker and more efficient. Same with the alveoli with the short diffusion distance. So alveoli are usually surrounded by these capillaries carrying blood. You can see it here with the red blood cells in it. And so there's a very short diffusion distance for oxygen to move from the lungs into the blood. So surface area, concentration gradient, diffusion distance, and temperature all come into play when we're talking about rates of diffusion. Okay. That was the first section. So, mini quiz. So if you all find the webinar chat and pop the answers to these questions in there. So question 1, is diffusion a passive process or an active process? So type A for passive process, B for active process. Really good, loads of A's coming in, fab. The correct answer is A, diffusion is a passive process. It doesn't use any energy. It's purely done from the natural movement of particles. Number 2, which of these does not affect the rate of diffusion? So A is concentration, B diffusion distance, C temperature, D sunlight, E surface area. Which of those doesn't affect the rate of diffusion? Good, loads of people saying D, fantastic, that's right. So sunlight doesn't affect the rate of diffusion, but concentration, diffusion distance, temperature, and surface area all do. We talked about those. And finally, question 3, particles move from an area of low concentration. Oh no. Let's just alter what I've written there. I've swapped low and high round. It should be an area of high concentration to an area of low concentration. This is described as A, moving up the concentration gradient, or B, moving down the concentration gradient. Good, loads of people putting B, despite my attempt at an inadvertent trick question. So when particles move from high to low, they go down the slide, down the concentration gradient. Fab. Okay, let's move on to our next mode of transport in cells. And that is osmosis. Osmosis is a special type of diffusion. So it's still a passive process, it doesn't take any energy, and it's still moving across cell membranes, but osmosis is particularly for water. So the official definition that you should write down in exams when you're asked is that water moves. So osmosis is when water moves from an area of high concentration to an area of low concentration across a partially permeable membrane, which is exactly what our cell membranes are that we talked about yesterday as part of cell biology. So all cells are surrounded by a partially permeable membrane, a cell membrane, that controls what enters and leaves the cell. Area of high concentration of water would be a dilute solution, and an area of low concentration of water would be a concentrated solution. Now, this can be quite difficult to get your head around, but the secret is to think of squash. So, if you've got a glass of squash and it's dilute, there's lots of water. So there's a high concentration of water. If you've got a concentrated solution of squash, there's more squash. You put a lot in there when you put the squash in the bottom of the glass, and then you filled it up with less water than you would need to if you'd made a dilute glass of squash. So always think about squash when you're thinking of concentrations of water. So a dilute solution has lots of water in it, a concentrated solution has less water, comparatively less water in it. Osmosis, like all of those other particles, will mean that water randomly moves towards that area of lower concentration of water, so into that more concentrated solution. So I've got a good diagram here. So you can see on one side, you've got a concentrated sugar solution, in this case, not squash, although squash is quite a lot of sugar, so I suppose you could think of it the same. So you've got lots more sugar particles in there, those little blue dots. They've rigged up a selectively permeable membrane in the middle of the beaker. And on the other side, you've got a much more dilute solution. So there's much fewer sugar particles, fewer of those blue dots. That means that there's more water, so there'll be a higher concentration of water. So concentrated solution has lots of sugar and less water. Dilute sugar solution has less sugar, more water. Okay, so when we think about the movement of water, water wants to move from the high concentration to the low concentration, down the concentration gradient. So it moves across the partially permeable membrane. Partially permeable means that things can go through it, so it has almost like little holes in it. Partially permeable is like a sieve. So the water moves across that membrane to the section of the beaker where there's a lower concentration of water. It's trying to balance it out. It's moving across. So that's osmosis. What we're going to talk about now is we're going to talk about more about those different kinds of concentration in solutions and how we describe them, and then we're going to apply that to cells. Okay. Describing different solutions when talking about osmosis. This comes up a lot in exams. It's well worth going through it over and over until it makes sense, and then practice. Do lots of practice questions talking about different types of solution. So the main function of osmosis is to try to keep the concentration of solutes, so things that are dissolved in water, inside the body cells at the same level. And we do this by moving the water about through osmosis. So we try and keep concentrations similar. This is done by a careful balance of movement water in and out of the cells. So, in these three diagrams, and we'll go through them in detail in a minute, so don't worry about looking at all of the writing now. The orange area is the cell, and the green area outside is outside the cell. And the dots are a solute, so something that's dissolved in water. So like the sugar in the slide before, or the squash when we were talking about squash in a glass. There are three ways of describing a solution compared to the cell, and this will come up a lot. So we use this language fairly regularly. So we're going to start on the left here with a hypotonic solution. Notice the way it's spelt, hypo. Hypo means low. So if the concentration of solutes outside the cell is lower than the concentration of solutes inside the cell, internal concentration, then we say that that solution outside the cell is hypotonic to the cell. Okay, so if we look at the diagram, we've got one little particle in solution outside the cell, and we've got lots and lots of the particles inside. So we have a higher concentration inside and a lower concentration outside. This means that outside the cell can be described as hypotonic, lower concentration, hypo means lower, to the cell. Okay, next one over. Isotonic solution. So if we have a look at this diagram, there's lots of dots outside the cell and there's lots of dots inside the cell. Concentration of solutes outside the cell, remember solutes are the things dissolved in the solution, is equal to the concentration of solutes inside the cell. They're perfectly balanced. Remember we were talking about how osmosis is always trying to rectify that balance. The solution is therefore described as isotonic to the cell. So iso in this case means the same. And finally, we move on to the next level, hypertonic solution. Hyper means more. In this diagram, we've got lots and lots of dots in the solution outside the cell. It's quite a concentrated solution outside the cell. It's a high concentration. And then inside the cell, we've got fewer dots. So there's a lower concentration of the particles inside the cell. That means that there's a higher concentration of water inside the cell, because there's more water filling the space around those particles of solute. So in this case, the concentration of solutes in the solution outside the cell is higher than in the inside the cell, so than the internal concentration. And therefore, we say that the solution is hypertonic to the cell. Okay, so hypotonic, low concentration outside the cell. Isotonic, the same. Hypertonic, higher. The concentration of solute is higher outside the cell. Okay, really worth thinking about, spending some time getting your head around. So don't worry if I've just explained that and you've gone, oh, don't understand that. She's talking about concentrations all over the place. That's okay. Take down the notes or take a screenshot of this slide and just go through it once more later, or answer some questions about it. Or ask me later and I'll go over it again. Okay. How does this relate to cells? So, plant and animal cells are always using osmosis, and they use it to try and keep their shape. So, but that means that you have to be quite careful with how much water goes in and out and how you're balancing your solutes, because obviously a cell only has a certain area, so you can put too much in or too little. I think this diagram is great. It's using those three terms that we just learned: hypotonic, isotonic, and hypertonic, and it shows you the results of those situations on plant and animal cells. So let's do animal cells first along the top, and let's start in the middle with an isotonic solution. Isotonic meant the same. So we have the same concentration of solute inside and outside the cell, and so water is moving in and out, maintaining that balance, doing what it should be doing, and the cell is normal. There's lots and lots of other things moving in and out as well, altering that concentration, so the water is continually moving to balance that out. In a hypotonic situation, and if you remember, in a hypotonic situation, there was a low concentration of solute outside the cell. That means there's a high concentration of water, because there's only one little solute and then lots and lots of water surrounding it. Whereas inside the cell, we have comparatively more solute and less water. So we have a lower concentration of water inside the cell. Therefore, water moves into the cell. If this isn't checked, the cell will fill with water and eventually burst. And we call that being lysed. Burst. In contrast, if you've got a hypertonic solution outside the cell, remember hypertonic means high, lots of solute, which means it's very concentrated and there's less water outside. Inside the cell, there's a lower concentration of solute, so a higher concentration of water. The water always wants to move down the concentration gradient, so it moves out of the cell. You can see H2O moving out there. That means that the cell will shrivel down. When the cell shrivels down, it can't perform the functions that it normally does. So both having a cell lyse or a cell shrivel can be really bad. And if it happens enough, then it can be deadly. So for example, if we get really dehydrated, eventually our cells shrivel and die. Okay. Plant cells are slightly different again. So for this one, we're going to work from left to right. So we're going to start with a hypotonic solution. Plant cells like to be plump. If you remember, they have a vacuole and they have a cell wall for keeping them plump. The vacuole that's full of cell sap comes into play an awful lot when you're talking about osmosis in plant cells. So the vacuole, because it's full of cell sap, it means that there's less, it means that that area of the cell that's taken up by that vacuole isn't full of water. That means that there's a lower concentration of water in plant cells generally than you would expect, because quite a lot of it is taken up by that vacuole. So in a hypotonic situation, so when the solution surrounding a plant cell is hypotonic to the cell, that means that there's only one little solute, so a low concentration of solute, but a high concentration of water outside the cell. That means that within the cell itself, you have more of those solutes and a vacuole. So there's a lot less water in the cell, very low concentration of water. So the water moves into the cell to keep it turgid. That's normal for a plant cell. Plant cells like to have water coming in. Okay. That's why those xylem cells that we talked about yesterday that transport water around the plant, they're very important. Plants need lots of water. If you have an isotonic solution, so your water outside the cell and the water inside the cell are at roughly the same concentration, you have roughly the same concentration of solutes as well, water is moving in and out. It's just moving around randomly, because remember, there's still that random movement of particles both ways, but in general, they'll move mostly in one direction. So You still have movement both ways, but it's roughly the same. And in that case, a plant cell is what we call flaccid. So it's not as plump as when it's turgid, heading towards shriveled. But and this isn't a normal state. It's normal for a plant cell to be turgid. If you have a hypertonic solution, so hypertonic solution means that there's lots, high, so lots of solute outside the cell, which means less water outside the cell. Within the cell, you've got less solute, but more water, so the water wants to move out of the cell. This means that the plant cell becomes shriveled, and we call that being plasmolyzed in a plant cell. Very, very bad for plant cells, because remember, they're used to being turgid, so if they shrivel, then they're much more likely to die. Interestingly, flaccid is, if you've ever seen plants that you've had on the side and you haven't watered them for a couple of days, all of the cells go flaccid and the plant wilts. So that's what happens when when plant cells don't get enough water. And eventually they'll shrivel completely, and then you get those really shriveled, dried plant cells when they're dead. Okay, so that's osmosis. Little mini quiz about osmosis, and then I'll go through some questions. So, ready on the chat box. Number 1, is osmosis an A, passive process, or B, active process? Okay, so type A for passive, B for active. Good, lots of A's. Yes, it's a passive process. This doesn't take any energy from the cell. It's all done on those concentrations across the membranes. Okay, fab. Question 2. If the concentration of solutes outside a cell is lower than the concentration of solutes inside the cell, the solution is described as A, hypertonic, B, hypotonic, C, isotonic. Think carefully about this. Concentration of solutes outside the cell is lower than the concentration of solutes inside the cell. Good, lots of B's coming in. Correct, hypotonic. Hypo means low. The concentration of solutes was lower. Remember, that means that the concentration of water is higher outside the cell than lower. You need to have those things in mind all the time. Question 3, which way will water move in the above situation? A, into the cell, B, out of the cell. It's hypotonic. And we just said that the water outside is of a higher concentration than inside, so the water will move into the cell. That would be A, into the cell, be the correct answer to 3. So you need to be really careful when you're thinking about these questions, when you're talking about the concentration of the solutes in relation to the concentration of water. So remember when we talked about the glass of squash at the beginning, high concentration of solutes, lower concentration of water. Lower concentration of solutes is more dilute, so there's more water. Okay, question 4. In an animal cell, what effect would this have? So water is going into the cell, out of control. Good, or puff up. Exactly. It might become lysed if it's really bad, then it might burst. And number 5, in a plant cell, what effect would this have? So we've got water coming into the cell. What's different about plant cells? Good, so Jacques, will be beneficial towards the plants. They like to be turgid, exactly, and that's normal for plant cells. Loads of people putting in turgid there, which is brilliant. That is normal for a plant cell. They like to be turgid. They like to be nice and plump and not wilting. Great. Okay, I'm going to do a quick scroll through some questions before we do our last How many people roughly are in these webinars? Depends. Today there's about 27 of us, I think. Sometimes there's more, sometimes there's less. An animal cell is hypotonic, puffy and fat because it has too much inside, and hypertonic is small and shriveled because it doesn't have enough. Exactly. That's why we have to drink water all the time. We need to keep all of our cells topped up at the right level, but not too much. Okay, I'll just scroll up through the chat because I think I saw a couple of questions in there as well. It's partially permeable, the same as semi-permeable, yes. And that means that stuff will pass through it, a bit like a sieve. So it's got little holes in it. Yes, so an animal cell, those cells that I described were red blood cells in the last diagram. Those are red blood cells that we were talking about there. And yes, they ideally are in an isotonic solution. So that's what they're always striving towards. Whereas plant cells like to be turgid, so they like to have water coming in. So you can die if you drink too much or too little water. You have to do this to a real extreme for it to be deadly. So we can survive about 3 days without water. But you'll know if you've gone a day without drinking water, you don't feel very good. You need water. You feel thirsty. That's the sign of dehydration. That's your cells saying, hey, I need some more water here, getting a bit shriveled. If you drink too much water, it's very difficult to drink too much water. Your body doesn't want to do it. And so I think our recommended water allowance, I think, is about 8 pints a day. Is that right? That might be wrong. So it's quite a few glasses of water, depending on the size of your glass. You would have to drink an awful lot more than that to make your cells burst. There are some drugs that can make you not feel the effects of drinking too much water. So there have been cases of people that have taken drugs, thought that they were thirsty, and their bodies haven't been able to switch off that thirsty feeling, so they've drunk too much water and then they have died, and that's because their cells rise. You can't die from lying in the bath for too long because your skin stops the water entering your body. Okay, so let's move on to our last type of transport. So far we've talked about diffusion and osmosis. They're passive processes. They're all about concentration gradients. They kind of go with the flow. But what happens when we need to move things fast and we need to move them against a concentration gradient? Then our cells have a process called active transport. And it's called active because it costs energy. So active transport is the movement of substances from an area of low concentration to an area of high concentration across a partially permeable membrane. So it's the opposite, it's going the opposite way. This involves energy from respiration. That's where the energy comes from, and often the use of transport membranes within transport proteins within a membrane. So our partially permeable membranes are cell membranes around a cell. They will let in and out the particles that go by diffusion. They tend to be very small particles like water, like glucose, like oxygen and carbon dioxide. But some of the things that need moving by active transport are quite big, and they need special proteins within the membrane to do that transport for them. So like a channel. Cells that respire a lot often need to use active transport. So we talked about cells that are specialized, that have lots of mitochondria, so they can do lots of respiration. And that's usually because those cells are specialized for actively transporting things. So root hair cells are a prime example. They're moving stuff from the soil into the plant. And water can move by osmosis, but there are other things that can't, that have to be actively transported. We'll talk about that in a sec. So nice diagram here. Got a solution here with a low amount of solute, high amount of solute inside. So this solution is hypotonic to the cell. We want to move that solute into the cell rather than naturally it would want to move out. It's a big particle, we need to move it in against the concentration gradient. So we use a transport protein and some energy to give that solute a push across the membrane. And there we have it moving in there in the second picture. So joining that big group. Oh, too quick. So why do we need active transport? Oh, let's just come across there. So we talked about root hair cells. So root hair cells actively transport mineral ions in the soil, e.g. nitrates. They transport them into the root of the plant to be used elsewhere in the plant. Some minerals like nitrate are often found in very dilute solutions. So they're very hypotonic to the solutions in the root hair cell. So active transport is needed to move them into the root hair cell against their concentration gradient. Root hair cells have lots of mitochondria to provide the energy for this. So here you've got the soil, we've got lots of the minerals and things inside the root hair cell, all being transported up to the plant slowly. But there's even less of those minerals in the soil. Soil is big, there's soil everywhere, and so there's lots of other particles in amongst the soil apart from the minerals that the plant needs, which means that it's very dilute if you think about it as a solution. Finally, glucose in animals is sometimes actively absorbed from the gut into the blood. Glucose is needed for respiration, so it's important to get as much as possible out of the gut where you're digesting. Concentration of glucose in the blood remains steady, which means that sometimes, as the blood moves along the intestine, this is higher than the gut concentration of glucose. Therefore, glucose must be moved against the concentration gradient. So glucose is one of those funny particles that can move by diffusion or active transport. And we talked about it before when we talked about the intestinal cells with the villi on them, lots of surface area for diffusion of glucose across into our blood. Well, as that blood is moving along, you've got different parts of your intestine that are digesting different bits of food. And so the concentration of glucose in the intestine goes up and down a bit. Blood comes by. Sometimes the blood, if you're getting towards the end of intestine, has quite a lot of glucose in it already, and it's in quite a narrow area in that capillary. So that means that it has comparatively quite a high concentration of glucose, and so it's better to actively transport that glucose out of the intestine and get it to the muscles as soon as you can. Okay, that's all you need to know about active transport for now. Mini quiz, last mini quiz, and then we'll do another round of questions again. So question 1 in the mini quiz, if you're ready on the chat function, is active transport an A, passive process, or B, active process? Type A or B. Ooh, a mixture there. Active transport is an active process. It requires energy. So B is the correct answer there. Question 2, active transport can be needed to move substances from very A, dilute conditions, or B, concentrated conditions. Good. Question 2 is dilute conditions. So in the soil, those nutrients were in very dilute conditions. There was only like one little nutrient and then lots and lots of all the other things in soil around it, which means it's very dilute for it. Good. Exactly, it is opposite to diffusion. That's exactly it. Okay. Quick questions about active transport. Someone's asked, is this like the difference between aerobic and anaerobic respiration? Yes and no. We're going to do more about respiration. I think next week we'll get to respiration, so tune in then and we'll talk about that more, anaerobic and aerobic respiration. Is semi-permeable membrane the same as a partially permeable membrane? Yes. Okay. I think that's everyone's questions. Let's go through some past paper questions so you can see how this knowledge might be used in an exam. If you do have any more questions, pop them in the question box and we'll have another look at them before the end. Okay, this one's slightly blurry. So question 1. This is a 1-mark question, but we still need to read it carefully. So underline the keywords in red. Substances can move into and out of cells. How does oxygen move into and out of cells? Okay, so we talked about this earlier in the lesson when we talked about how oxygen moves from the lungs to the blood, and then from the blood into the cells of the body. And it does that by diffusion, not digestion and not photosynthesis. It's 1 mark, so all we need to do is draw the ring around diffusion. So 13, this is a nice diagram here. Diagram 1 shows the percentage concentration of oxygen in 3 cells, A, B, and C. So let's have a good look at this diagram. Cell A here, at the top, has 5% oxygen, fairly low. Cell B in the middle, 20% oxygen, so quite a lot higher, 4 times higher than cell A. And then cell C has 10% oxygen. Oxygen can move from cell to cell by diffusion. Into which cell, A, B, or C, will oxygen move the fastest? Remember, from diffusion, we're moving from a high to a low concentration. So into which cell will oxygen move the fastest? Good, somebody pop it into the chat there. A. Good. The answer is A, because A has the lowest concentration of oxygen. 1 mark there. How does water move into and out of cells? Draw a ring around 1 answer. Breathing, osmosis, or respiration? Loads of people saying osmosis there, brilliant. It's not respiration, that's a process, it's not breathing, also a process. Osmosis is how water moves between cells. 1 mark there. Three different cells, P, Q, and R this time, rather than A, B, and C. Percentage concentration of sugar solution in each cell. So cell P has 1% sugar solution. That means that it's very dilute. 1% sugar solution, there's only 1 molecule of sugar in there for every 100 molecules. So the rest is all water. So it's a high concentration of water, low concentration of sugar. 2% sugar solution in cell Q and 3 in cell R. They're all pretty low percentages of our solute in this case, which is sugar. They're very dilute. If they were a cup of squash, they wouldn't be very nice, they'd be very watery. Water can move from cell to cell. Into which cell, P, Q, or R, will water move the fastest? Think about this carefully. Some difficulties in the chat here. So think carefully about this. Water wants to move towards, so water wants to move down its concentration gradient. It wants to go from a high concentration to a low concentration. Of which of those cells, which cell has the lowest water concentration? Not sugar, water. Good. There's a few people catching it. There we are. It is cell R. It's good to do lots and lots of these questions to really get in your head the difference between the solute, the concentration of the solute, and the concentration of the water. It's really important. So in this case, the solute was sugar. So sugar had the highest concentration in cell R. Therefore, it had the lowest concentration of water. Water wants to move to the lowest concentration, down the slide, down the concentration gradient. So it moves to cell R. Good. Another couple of nice, simple, quick questions here. 2 marks for these. Substances can move into cells and out of cells. Okay. A, draw a ring around the correct answer to complete each sentence. So water moves into cells and out of cells by active transport, osmosis, or reabsorption. Good. Osmosis there. And then finally, the water moves through a freely permeable, non-permeable, or partially permeable membrane. Good. Partially permeable. That means there are some holes in it. A freely permeable membrane would mean that it had lots and lots of really big holes. Freely permeable, everything could pass through it all the time. Non-permeable would mean that it's completely sealed. Partially permeable means that it has got holes in here and there, so it can let some things in and out, but not freely. Good. Students put plant cells into 2 different strengths of sugar solution, A and B. The diagram below shows what the cells look like after 1 hour. So the cell in sugar solution A after 1 hour looks pretty good. Remember, plant cells like to be turgid. They like to have lots of water in them. You can see the cell wall is nice and plump, the vacuole's nice and plump. We've got a good cytoplasm there. Whereas the cell in sugar solution B, after 1 hour, has a bit of a shriveled cell wall. The vacuole's much smaller. There's space between the cytoplasm and the cell wall, so the cytoplasm's drawn right in. It's really shriveled. Describe 2 ways in which the cell in sugar solution B is different from the cell in sugar solution A. And if you can remember everything I just said in the last minute, I just told you the answer. Good, yes, has less water in it. That's really good. You can see that the cell wall is a bit shriveled. Yeah, nucleus isn't touching the cell wall. Cytoplasm is drawn away from the cell wall as well. Much less water. Yeah, that's really good. A student put red blood cells into water. Suggest what would happen to the cells. So if you put those red blood cells into water, pure water, bear in mind normally red blood cells are surrounded by things other than water as well. If you put that red blood cell into pure water, got a high concentration of water outside the cell, low concentration of water inside the cell. The solution outside is hypotonic to the solution inside. What's going to happen to the cells? Yes, it would become plump and eventually burst. Really good. Lysed, exactly. That's the key word there. Would become lysed. Good. Let's do a final skip through here for any questions. Does it have to be across a membrane? Yes, because to move from one area to another, it would be across a membrane. Does active transport only transport from dilute into concentrated? Good question. Strictly, no. You could actively transport particles from any concentration to any concentration because you're using that energy to give them a push. But in general, we like to make life easy. So if we can do it by diffusion, we'll do it by diffusion. So it's only when the particles are particularly big or in a particularly dilute solution that we tend to use active transport. There are exceptions, but because obviously there's some conditions that change and things, but most of the time we only use active transport and we need it in order to conserve our energy. Someone asking, when will we learn ecology? That comes quite a lot later on in the specification, so be a couple of weeks' time, I'd have thought. Okay. That's all the questions. Then I think we're nearly there. So there'll be more biology lessons next week. The topics on those, we're still kind of finalizing them at the moment, but I think we'll be moving towards respiration and photosynthesis. We might do a bit of maths stuff as well, so keep an eye on the timetable. That will have information about what topics we're going to do. And make sure that you attend if you want to. In the meantime, if you want to find some past questions to do, then have a look on the My Tutor website. We have lots of past questions and answers to questions on there. You can also have a look at BBC Bite Size. That's a really good resource for GCSE Biology as well. And if you're really struggling, remember that My Tutor has lots and lots of tutors that will tutor on a one-to-one basis. So you can find those on the website. And do have a think about whether that might be helpful. And you can find all the information on that on the My Tutor website as well. So I think, unless there's anything else to say, Bola, I think we're there. So Bola from My Tutor here is here as well, in case I have any technical difficulties or anything like that. Anything else?
No, I think you've covered it all. Thank you very much, Charlotte, for a great lesson as per usual. And thank you all for joining us. Have a lovely afternoon, guys. Goodbye.
Bye, everyone. See you next week.